Thanks to the organisers for inviting me to talk. I'm Toby and I work at UCL, the Institute of Child Health, in the Genetics and Genomic Medicine department. And I'm in a lab which is in a set of labs which study a series of diseases known as ciliopathies. And they are a set of developmental diseases, and as the name would suggest, they result as from problems with the cilia in the cells, be it motile or primary cilia. And I study Jeune syndrome, which is a skeletal ciliopathy. And it's characterized by a very prominent thoracic dysplasia, as you can see here. And this causes severe respiratory problems, and as a result, the actual perinatal mortality rates are quite high. And even those patients that go on to survive early childhood, they run the risk of developing other ciliopathy symptoms such as renal cysts and visual problems. So mutations in the protein IFT80 or the gene IFT80, missense mutations, cause Jeune syndrome. And IFT80 stands for intraflagellar transport protein, and it's responsible for anterior grade transport of cargo from the base of the cilia to the tip of the cilia. Very little is known how the mutations translate into the disease, and so I'm just going to talk briefly how we've employed CRISPR in just the humble immortalized cell line to kind of understand the processes of ciliogenesis in Jeune syndrome. So the format of the talk, first I'm going to talk, like I said, about these cell lines. These are intramedullary collecting duct cells, and I'll talk about how we engineered some knockout cell lines with CRISPR. The CRISPR's kind of surpassed usefulness in our lab over RNA knockdown techniques, but I'm not going to go into too much detail about that. As a result of using the CRISPR, we believe we got some unanticipated phenotypes by knocking out the protein instead of knocking it down. And then briefly, I just want to tell you about other things that are going in the lab since we've been embracing the technology. So the best way for me to tell you how we've used CRISPR is just to address a question that's kind of been omitted from the literature. We don't know what these mutations do, and so for illustration purposes, I'm just going to say the hypothesis is that because all patients with IFT-associated Jeune syndrome have at least just one missense mutation, we can hypothesize that maybe these mutations are hypomorphic, as in they're not a total loss of function, they have some residual function. So this figure is actually lifted from a multi-award-winning poster by one of our PhD students with her permission, and because it really concisely illustrates the methods that we use. And it's simple. We do a co-transfection with the CRISPR plasmid PX330 with GFP. I'm aware that there's now plasmids with GFP in. And then we subject our cells, the IMCD3s, to FACS sorting, and we do two sorts. We do the group sort for a diagnostic to see how well the guide has cut, and then the single cell sort to establish the monoclonal cell line. So I'm sure you're all quite familiar with that method. And this is just a schematic to show how we went about knocking out the protein. So it's quite a large gene, and we went for multiple high-scoring guides, multiple guides on early exons, but other exons as well. And the reason why I use multiple guides is obviously to address the whole off-target issues. And then we subject this to the T7 nuclease assay. And so here you can see the blue lines, the PCR fragment, and the black lines are the anticipated T7 nuclease fragments that we got. And as you can see here, so these are two guides we used for one exon and other exons. So and also there was only one splice variant with this protein, so we were confident that this strategy would knock out the protein. So excitingly, we found that sorry, I've really brought out the color of these because I've had problems before with my immunofluorescence, but we found that in the knockouts, these are the genotyping of the compound indels that we got, that there were no cilia, so this was quite exciting. This was very useful for also finding further cell lines. And this is an ARL13B, it's a marker for cilia, but we also use microtubule staining. But the reason why I'm showing you ARL13B is because, well, you'll see in a bit, we use it for assessing ciliogenesis. And also, if you see there's actually a basal body and centriole defects, which I'm going to talk about in a bit. So that was quite exciting. And then what we did was, to confirm the phenotype, we rescued it with IFT80 and a tagged version of IFT80, which was great because we've not really visualized IFT80 before, and it took a while to find a configuration with the GFP tag that expressed. And so as you can see here, the IFT80 in the green cells, in our knockout cells, localizes to the cilia and also to the basal body. You can't see it very well. And again, we got the ARL13B stain here. Now what you can see is that the transfection efficiency of these cells is not 100%, so it's kind of nice to see the knockout amongst the ones that have been rescued. So we think we got a nice model that we can now transiently reintroduce the mutants from Jeune syndrome into, but there was one hurdle, and I think there's some cilia people in the crowd, and they'll know how annoying it is to assay ciliogenesis. There's a lot of cilia. This is a wild type. There's a lot of variation, and there's a lot of parameters you want to measure. So we consulted our imaging specialist in the department, Dale Moulding, and he helped me come up with some programs, a macro to kind of assess ciliogenesis, and with some cool particle analysis, he managed to single out the cilia as particles, and he put some skeleton frame over it, and from this we can gather the length of the cilia. So this is why I'm showing the ARL13B stain, of course. We check to see that the microtubules are there as well. And then we can confirm it's cilia by seeing that it goes to the basal body. So we have our model, and we have our way of acquiring our data. And so we jumped in and we transiently transfected the mutants. We didn't obviously go for homology-directed repair approach because we had multiple mutants that we wanted to test. The reason why we wanted to test multiple mutants is because these ciliopathies have such heterogeneous symptoms, and it'd be interesting to look at individual mutants. They were scattered over the protein, and to our surprise, we found, well, when we looked addressing the hypothesis, we found that all the mutants rescue ciliogenesis. And now, of course, I'm addressing a very simple hypothesis here. So we're showing that, firstly, that no IFT80 ciliogenesis and Jeune syndrome missense mutations rescue cilia, and so we can confirm that it's not due to a complete loss of function. But in reality, what we're doing is we're assessing more complicated things like the trafficking of the protein. Dale Moulding has helped us devise some more intricate programs to look at the trafficking of IFT80 and trafficking of other cilia markers in these knockout cells, which has the benefit over knockdowns because there is no residual wild type there. Also, I'm going to address the off-target consideration, seeing it's a CRISPR conference. So like I said, we used multiple high-scoring guides. The same phenotype was seen in multiple guides, different off-target effects. Any phenotype that I suppose is exclusive to one guide, and we can dismiss. But we were lucky. I think our phenotype was very obvious. You knock it out, no cilia. And we found very robust rescuing, which was great and very exciting when that happened. And also, this is quite interesting. We also, in the process of genotyping, we found in-frame deletions, and I think this represented quite a nice intermediate. I didn't get any wild type and then knockout, but here you can see that there are still cilia, but they're shorter. So we think this is quite a nice intermediate to show. And although these mutations are specific to one guide, and you might want to be careful with the interpretation to see whether the shorting is due to that guide in particular. What we could do is those knockout cells came to the rescue again, and we've actually introduced these mutations to show that this mutation actually doesn't rescue cilia. So this is quite a nice model. And the significance of this is now is that we have a model that we're not only testing the pathogenic mutations, but we're testing structural mutations, looking at the domains, and we're in collaboration with a structural lab in Germany who are interested to see this protein just in the general scheme of ciliogenesis. So that's that. And also, this is an unanticipated phenotype that's found. I mentioned basal bodies. In the knockout cells, we found that it's increase in this central splitting, which don't do too much about, but it was brought to my attention at another conference. And in the rescue cells, you see these centrioles are unified, the basal body, and we quantified this, and we found in the IMCD cells that the IFT actually rescues this phenotype. So you see the number of these cells is decreased. And so we asked whether this is part of the disease mechanism, but preliminary data suggests it's not. But nevertheless, we can go and we can probe the function of IFT80 now in these cells and see, because this is not the only cell cycle defect we're finding quite a few. It's interesting. So I'm just going to quickly talk briefly. I think I've rushed through it, but these cells are a polarized cell, and so therefore they form spheroids, which are shown here. And typically this assay was used with RNAi, so you're knocking down, you can't get rid of all the protein. But now we've produced in our lab, we're producing multiple IMCD3 lines that are knockout. And then we can assay them in the spheroid cultures, which are obviously great for things like screening drugs and stuff. And as you can see, in the wild type, the cilia are nicely orientated inwards. And in IFT80, surprisingly, they still form spheroids. You see the absence of cilia. These are not cilia, these are actually mid-bodies. And so you can kind of conclude maybe that IFT80 and cilia aren't involved in this PCP, this planar cell polarity, which isn't too far removed from what's reported in the literature. What we did find, and I'm having some people help me with this, is they're assaying the increase in these mid-bodies, which is again some cellular defects that we're looking into. And finally, we actually are anticipating the arrival of some mice, so we've got 2 mutants coming in, and they all, again, have been used with CRISPR because obviously now the way that they can make the mice seems to be more efficient, and this is the strategy that they used. And what's a really nice approach is that in making these mice, we should be getting a knockout as well as a knock-in, and that's useful because it's on the same isogenic background. We are keenly anticipating a phenotype with this. And obviously there's a lot of gene therapy emphasis here, but ciliopathies are certainly candidate diseases for correction with, but they're monogenic, so the correction with CRISPR-Cas9. I suppose the added complication with things like Jeune syndrome is it has to be like in utero gene therapy. Yeah, I think this is the direction that the lab wants to go in, well, if Dagan and my boss wants to go in. Anyway, so I'm just going to thank the people here. Dagan, my boss, for letting me work on the project, and Grace for giving me the figures. Aphrodite, who was a master student in our lab and helped me with the CRISPR when we were setting it up. Dale, obviously, for the imaging, and I, Stephanie, for the facts, and Tiago and Katia for introducing me to 3D cell culture. That's it. Oh, that's nice.